Okay, in this session, we're going to look at A2 respiration for CCEA. First part of this is looking at the structure of mitochondria. So here we can see a normal eukaryotic cell, nucleus, etc. But you can see here the structure of or where the mitochondria are found within the cytoplasm of the cell. In terms of the detailed structure of the mitochondria, they have an outer membrane, which you can see around the outside. The inner membrane is highly folded with these cristae or crista, and there is also a space between the outer and inner membrane known as the intermembrane space. And then the center of the mitochondria, the sort of cytoplasm of the mitochondria, is known as the matrix. Okay, the cristae, these foldings that you have in the inner mitochondrial matrix, have these little stalked particles, which we'll take a look at in more detail later on, are known as Atp Synthase, and they are little proteins involved with the synthesis of ATP during the electron transport chain or oxidative phosphorylation. Okay, again, sort of detailed structure there, outer membrane, intermembrane space, these foldings or cristae with the little stalked particles that you can see, and then the matrix is the bit in the center. Okay, the next part of this is looking at the structure of ATP. So ATP, known as adenosine triose phosphate, and as that suggests, it's got 3 phosphate groups. You don't need to know this detailed structure. That is the type of structure that you need to know there. So you've got a ribose sugar, 5-carbon sugar, you've got an adenine group, and 3 phosphates make up ATP. Okay, the energy, it's sort of known as the energy currency within cells, and the energy is stored between these bonds, between the phosphates. So when these bonds are broken, then it releases energy. So ATP can be broken down into ADP, adenosine diphosphate, as it suggests it lost one of its phosphate groups. So energy has been released as that bond has been broken. Okay, so ATP is sort of your fully charged battery. And as I say, when that third phosphate is broken off, it's sort of known as inorganic phosphate. So that would be your ATP being broken down into ADP plus phosphate or plus PI, inorganic phosphate. And that releases energy, which then can be used in body processes like muscle contraction, active transport, nerve contraction or nerve impulses, etc. That uses up the charge within the battery. Okay, the overview of respiration, it's split down into, I suppose, 4 stages. We've got glycolysis is the first stage, which happens in the cytosol and cytoplasm of the cell, releases a small amount of ATP. Then you've got the products of glycolysis going through the link reaction and the Krebs cycle or the citric acid cycle within the matrix of the mitochondria. Again, that releases a small amount of ATP. And then most ATP is synthesized or produced from what they call oxidative phosphorylation, which occurs on the inner mitochondrial membrane involving those stalked particles on the cristae. Okay, in terms of glycolysis, the first stage of respiration, it doesn't require oxygen. So glycolysis occurs in aerobic and anaerobic respiration. As I've suggested, it occurs in the cytoplasm or the cytosol of the cell. We begin that reaction with glucose, which is quite an unreactive molecule. It requires a bit of a kick up the backside to make it a bit more reactive. So some ATP is used and some ATP, a small amount of ATP is synthesized, as we'll see in a moment. You also have these coenzymes called NAD, which basically pick up hydrogens and bring them somewhere else. Okay, so they pick up the hydrogens. You have dehydrogenation going on here with glycolysis. So it loses hydrogens, which are picked up by NAD, and that becomes reduced NAD or NADH. And then what we have left is 2 3-carbon molecules of pyruvate. Okay, so as I said, glycolysis is a very unreactive molecule. It requires the breakdown of ATP or hydrolysis of ATP to make it more reactive. So you have ATP being broken down, the phosphate combines with the glucose to produce hexose phosphate. So these are both 6-carbon molecules. Another molecule of glucose is used to produce fructose biphosphate. So that's a bit like your 6 carbons with a phosphate stuck on each end of it. That is a bit more reactive and it automatically splits down into 2 3-carbon triose phosphates. They go through a series of reactions involving the loss of hydrogen, so dehydrogenation, and the synthesis of ATP, so some ATP being produced there, a phosphorylation occurring. You have 2 ATP produced per triose phosphate, and then that leaves 2 molecules of pyruvate, both being 3 carbons. Okay, so they talk about the net gain of ATP as 2 ATPs from pyruvate. So we've used up 2. We've got 2 produced here at each stage, so 4 produced, but because we've used up 2, then we have a net gain of 2 ATPs. Okay, and that's seen in this flow diagram as well. So you've got the glucose phosphate, so it's using that phosphate from the breakdown of this ATP molecule. Then you have your fructose phosphate, fructose biphosphate, so another ATP being hydrolyzed or broken down. Then your triose phosphates, series of reactions producing a net gain of 2 ATPs and 2 reduced NADs and leaving pyruvate. Okay, that pyruvate then moves into the matrix or into the mitochondria, and it goes through what we call the link reaction. Okay, so within the matrix, within the mitochondria, you have a molecule, or these molecules of what they call coenzyme A, okay, or CoA, floating around within the soup of the matrix. Your pyruvate goes through some decarboxylation, so carbon dioxide is given off, so this 3-carbon becomes a 2-carbon molecule. It also goes through dehydrogenation, so loss of hydrogen, which again is picked up by this coenzyme NAD, okay, to become reduced NAD or NADH. And as I say, it combines with coenzyme A to produce a molecule called acetyl coenzyme A, which is a 2-carbon molecule. Again, that just showing you pyruvate losing carbon dioxide to produce acetate, losing hydrogens, reduced NAD produced. Your coenzyme A floating around within the matrix then combines with that acetate to produce acetyl coenzyme A. That reaction, or that substance, acetyl coenzyme A, is then used in what we call the Krebs cycle or citric acid cycle. Okay, so it loses its coenzyme A, which is recycled back to the link for another link reaction. That 2-carbon molecule combines with a 4-carbon molecule floating around within the matrix called oxaloacetate. Those combine to produce a 6-carbon molecule called citrate, hence why it's sometimes called the citric acid cycle. It goes through a series of reactions, including decarboxylation and dehydrogenation. So carbon dioxide being lost, again, the hydrogens picked up by NAD to form reduced NAD. That leaves a 5-carbon molecule called oxoglutarate. Again, it goes through decarboxylation, dehydrogenation. There's enough energy there to synthesize 1 molecule of ATP. So it becomes a 4-carbon molecule. That goes through some more reactions here, including dehydrogenation. In this case, the hydrogens are picked up by a different coenzyme called FAD, and it becomes reduced FAD. You've also got some more reduced NADs produced from this, and then it recycles your 4-carbon oxaloacetate. Okay, so the products from the Krebs cycle, I suppose you could say you've got your 3 reduced NADs, so 1, 2, 3, 1 reduced FAD per Krebs cycle. Okay, 1 molecule of ATP produced from the Krebs cycle, and then you've waste gas in terms of carbon dioxide. Okay, so this is per Krebs cycle. So if you think about that, there'll be 2 Krebs cycles for each molecule of glucose, because you have 2 pyruvates, 2 link reactions, and therefore 2 Krebs cycles. So you have per glucose molecule 3 reduced NADs from the Krebs cycle and 2 reduced FADs from each Krebs cycle. Okay, now just to revisit these stop particles that you have on the inner membrane, as I said before, they're called ATP synthase. So you can see here that stopped particle passing from the inner membrane space into the matrix. That's a little enzyme called ATP synthase. It allows hydrogen ions to move through. As they move through, this sort of turbine at the top turns, and it combines ADP with phosphate to synthesize ATP. Okay, and this occurs in what we call the final stage is oxidative phosphorylation. Now this looks a bit scary. To just go through slowly, we have our reduced NADs and reduced FADs, which have come from glycolysis, the link reaction in the Krebs cycle. Okay, here's our inner membrane. Okay, and this bit at the bottom is our intermembrane space. So the matrix is up here where these are produced from the Krebs cycle. Intermembrane space is leading from the intermembrane space here into the matrix. So there's your little stopped particle or your ATP synthase. Within the intermembrane, you've got what we call electron carriers. Okay, so cytochromes, coenzyme Q, etc., flavoproteins you can see here. What happens is our reduced NADs and our reduced FADs dissociate or give up their hydrogens. Okay, and the NADs and the FADs are then recycled and can be used up again in glycolysis and Krebs cycle and link reaction. The hydrogens split into electrons and hydrogen ions or protons. Okay, and when they're dropped off at this flavoprotein molecule, those electrons are at very high energy levels. Okay, and as they're passed along these electron carriers, some of that energy is used to transport or pump these hydrogen ions from the matrix or the mitochondria into the intermembrane space. So we get a buildup of hydrogen ions in the intermembrane space. Okay, so that forms a concentration gradient, which then allows these hydrogens to pass back through into the matrix through our ATP synthase molecule. As they move through, ADP plus phosphates are synthesized or joined together to form ATP. Okay. As the electrons are passed down the electron carriers, the final electron acceptor is oxygen, hence the name oxidative phosphorylation. So oxygen picks up these electrons, it combines them with some of the hydrogens floating around within the soup of our matrix, and it synthesizes water, which is again one of our sort of waste products from respiration. Okay, and we can see that from this little animation.
In the mitochondrion, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, and that can be seen in this other diagram. It's just slightly the other way around. So we have our reduced NADs and reduced FADs dropping off our hydrogens, those splitting into electrons, which are passed through the electron carriers, picked up by the final electron acceptor oxygen to produce water. The hydrogen ions are pumped through into the intermembrane space, building up this concentration gradient. As they move back through, then that synthesizes molecules of ATP. Okay, and in terms of the numbers of ATP molecules produced, what they reckon is that for every one reduced NAD molecule, it will synthesize 3 ATPs, and every reduced FAD molecule, it will synthesize 2 FADs. So if we think about it, you have 2 reduced NADs produced from glycolysis. You will have, excuse me, another 2 per for the link reaction, so 1 for each link reaction. Remember, you have 2 pyruvates. You have 3 NADs produced per Krebs cycle, so that's 6 from here. So that's a total of 6 plus 2 is 10. So 10 would be converted into 30 ATPs in the electron transport chain. You'll have 1 reduced FAD per Krebs cycle, so 2 Krebs cycles in total, so 2 reduced FADs. Each of those producing 2 ATPs will give us a total of 4. So the electron transport chain will synthesize 34 ATPs. You've got a net gain of 2 from glycolysis. You have 1 for each Krebs cycle, so that's another 2. So 34, 36, 38 ATPs per glucose molecule. Okay, so 38 ATPs per glucose. There's just another little bit of an overview for where these reactions take place. So glycolysis is outside the mitochondria in the cytosol or cytoplasm. Then that pyruvate molecule moves into the matrix, your link reaction occurring in the matrix, Krebs cycle occurring in the matrix, and then oxidative phosphorylation using oxygen, producing water as a byproduct, is in the inner membrane. Okay, so those cristae on the inner membrane. Okay, glucose is not the only molecule that can be used for respiration. It's the main one. So glucose can be used. It's less reactions, I suppose, for glucose to be used than some of the others, and it'll form some ATP, more molecules of ATP. Lipids can also be used. So fats, if you like, can be broken down into fatty acids and glycerol. Glycerol can be used as a substitute in glycolysis. Fatty acids can be used as a substitute for acetyl. So those long hydrocarbon chains can be split into 2 carbon chains, which can be used as a substitute for acetyl, coenzyme A in the link reaction. Proteins can be broken down into amino acids, which again can be used as a substitute for pyruvate or for acetyl coenzyme A, or as one of the intermediates in the Krebs cycle. So just other substrates can be used. Okay, so that's aerobic respiration. Anaerobic respiration also occurs when oxygen is not present. Okay, and that is generally glycolysis plus a little bit more. Okay, so glycolysis can still occur. So you've got your glucose, triphosphate, pyruvate occurring in the cytoplasm or cytosol of the cell. There's a net gain of your 2 ATP molecules, so a small amount of ATP is produced from anaerobic respiration. This is anaerobic respiration in mammals. So your pyruvate in muscles is broken down by an enzyme called lactate dehydrogenase to produce lactate. Okay, now if you think your reduced NADs will still be produced from glycolysis, then those hydrogens from your reduced NAD are then used to reduce pyruvate down into lactate. Okay, and the NAD is recycled. All right, so this next bit shows that. So your glucose being broken down into pyruvate, small amount of ATP, net gain of 2. Your dehydrogenation of glucose produces reduced NAD. and then those hydrogens are used to reduce pyruvate down into lactic acid or lactate. Okay, now if you think about mammals anaerobically respiring, tends to happen in sprinting or weightlifting. So you're not using oxygen, you're building up that lactate in muscles, which is a toxic chemical. It requires to be broken down after, so when oxygen is present again, and the amount of oxygen to break down that lactic acid or that lactate is known as the oxygen debt. Okay, so that lactate needs to be broken down because it's toxic. The amount of oxygen required to do that is called the oxygen debt. Anaerobic respiration also occurs in plants, okay, mainly in yeast, and it can be used to produce ethanol or alcohol. Okay, so glycolysis again occurs, small net gain of 2 ATPs, your reduced NADs are produced again. That hydrogen again is used to reduce pyruvate down into, in this case, ethanol, but this also involves dehydrogenation, so some carbon dioxide being released. And again, with yeast, that can be used, or I suppose in terms of bread production, it causes the bread to rise, but it causes the production of alcohol as well. And that shows, this next bit shows again, glucose to pyruvate is glycolysis. That pyruvate loses carbon dioxide, decarboxylation, and then the hydrogens from the reduced NAD are used to reduce this down into our alcohol, which then can be used in alcohol production. Okay, the last bit of this then is looking at the respiratory quotient, or RQ, and that's an equation which is used to work out what substrate is being used for respiration. So in other words, what the input is. Is it glucose? Is it protein? Is it fat? RQ is generally the volume or the number of molecules of carbon dioxide produced divided by the volume of oxygen or the number of molecules of oxygen consumed. Okay, so if you take your word equation for respiration, you've got glucose there, requires 6 oxygen molecules, and that produces 6 carbon dioxide molecules and 6 water molecules. So in terms of numbers of carbon dioxide molecules and oxygen molecules, it's 6 divided by 6, which gives you an RQ of 1. Okay, and an RQ of 1 is the typical value for carbohydrate being used. Another example here is using aloic acid, which is a fatty acid. Okay, it requires 51 oxygens to react with to break it down into 36 carbon dioxides plus 34 oxygens. So your RQ is carbon dioxide divided by oxygen. So it'll be 36 divided by 51 gives you an RQ of 0.7. And again, that's a typical value for lipids or fats. You can see your carbohydrates here is 1, fats or lipids are 0.7, and protein has a typical value of 0.9. Okay, now if you did an RQ and an RQ equation, you got 0.8, it would be a mixture of fats and proteins. Okay, the other one you need to know about is that if the RQ is more than 1, then that means there's less oxygen, excuse me, less oxygen used than carbon dioxide produced. So that's an example, or that's a typical value, anything that's more than 1 of anaerobic respiration. But there's also some aerobic respiration here because there must be some oxygen being consumed. Okay, that can be worked out using something called a respirometer. Okay, so in this example, you've got your respiring organisms here. They use up oxygen and they produce carbon dioxide. We have potassium hydroxide in the bottom here, which absorbs the carbon dioxide. And then that causes this level to move up. So that would be the amount of oxygen consumed. Okay. If you wanted to work out the amount of carbon dioxide produced, you would replace this potassium hydroxide with water. So then you would see where this level moves to. If the level stays where it is, then it's the same amount of oxygen. If it moves up the way, then it would be less oxygen would be consumed. If it moves down the way, or sorry, less carbon dioxide. If it moves down the way, then it would be more carbon dioxide would be produced. Okay, and you can use a simple respirometer, to do this as well. Either have your potassium hydroxide or your water. You work out how much it's moved with potassium hydroxide. That would give you the amount of oxygen consumed. Replace it with water, depending on which way it moves. That will give you the volume of carbon dioxide produced. And the same with this one. You can use your respiring tissue. You have your potassium hydroxide in here, absorbing the carbon dioxide, which causes this level to move up. And then replace the potassium hydroxide with water. Again, depending on which way it moves, then that will give you the idea of the amount of carbon dioxide produced. Hopefully that helps with A2 respiration.